All right, sorry, my computer crashed. Thank you for all still being here. We will see if we can continue on with the video. I think it's almost over. So let me see here. Thank you guys for all hanging out. I appreciate it. Okay. I don't know where. Oh, come on. Sorry for the ad. Hold
By
on.
2050, there will be more plastic than fish in the oceans. There are 18.
Don't know where we stopped.
It's how we're trying back that may help you remember the name of this
we'll just start
phase
here.
called anaphase. After this, it's just a matter of using all that genetic material to rebuild so that the copied genetic material has all of the accoutrements of home. In the last phase, telophase, each of the new cell structures are reconstructed. First, the nuclear membrane reforms, the nucleoli form within them, and the chromosomes relax back into chromatin. Then a little crease forms between the two new cells, which marks the beginning of the final split. That division between the two new cells is called cleavage, and all that's left is to make a clean break. This is done by cytokinesis, literally cell movement, by which the two new nuclei move apart from each other and the cells separate. We now have two new cells, each with a full set of 46 chromosomes. These clones are called the daughter cells of the original cell, and like identical twins, they are genetic copies of each other, and also of their parents. But of course, that's obviously not the case for you. Even if you are an identical twin, shout out identical twins! See me in the comments. While you kind of are a clone of your sibling, you are not a clone of your parents. Instead, half DNA in each of your cells is from your mom and half is from your dad. To understand why that is, we have to understand how eggs and sperm are formed, and that is meiosis. And that's what we're going to be talking about next week on Crash Course. Until then, you can just watch this video over and over again, or you can just watch the bits.
Okay, so hopefully that was helpful. We'll probably watch a mitosis video too, not today, but we're going to talk about mitosis next. Are you able to see my PowerPoint screen? Anybody? Can you see the PowerPoint?
No.
No. Okay, thank you. Let me see what I can do. Just takes me a minute to figure out how the heck to do this. Screen sharing. Okay, there we go. Okay, you should be able to see it now. Can you guys see it?
Yeah.
Thank you. I appreciate that. Okay, so we've gone through mitosis. And remember, like, the ultimate goal of mitosis is to create two genetically identical offspring. Okay, two genetically identical cells. Genetically identical chromosomes split in two new cells. Bam. But how are you able to get one from two? Something to think about. So most of the cells in your body, except for in the gonads, are diploid. They have two copies of each chromosome, one copy from mom, one copy from dad. If your cells are diploid, how are you able to reproduce without doubling the number of chromosomes in the new cell? Anybody? Maybe I should make this an attendance question. It is a good question. So if our cells are diploid, how are you able to reproduce without doubling the amount of chromosomes in your offspring? This is the crux of meiosis. Okay, so gametes are the answer to this solution. So to prevent the doubling of chromosomes, sexually reproducing organisms have to make cells that have a single set of chromosomes. So they have to have a way to divide and make cells that have half the number of chromosomes that the parent cell had. So in sex cells, In the gonads, gametes, or we also call them sex cells, are haploid. They have half the genetic information that the parent cells did. They only have one copy of each chromosome. So male gametes are called sperm, and that's not just for humans, it's animals, even plants. Pollen is sperm. Yeah. Female gametes are called eggs or oocytes. Okay, so one pair of chromosomes makes you male or female, the X and Y chromosome. I think everybody's pretty familiar with that. So there are 46 pairs of chromosomes in your body, or sorry, 46 chromosomes. They're arranged into 23 homologous pairs. So homologous, that prefix homo refers to same. So that means if you have 23 homologous pairs, you have 23 pairs that are 23 pairs, and the pairs are the same. 23 from each parent. And we'll talk more about that in a minute. Of those 23 pairs, 22 pairs are called autosomes. And the prefix auto refers to self. So self or body. And these would be all of the chromosomes that are involved with the functions of your body. Okay, only one pair is involved with sex determination, and those are your sex chromosomes. Females have 2 X chromosomes. Males have an X and a Y chromosome. And by the way, men, you are the one who determines the sex of your offspring, because your offspring will either carry an X chromosome or a Y chromosome. So it's your sperm that actually determine the sex of the offspring. A little bit more on that later. Okay. You can organize chromosomes into homologous pairs. This organization of chromosomes in this picture you see here is called a karyotype. And it is photographic, and it helps kind of see the different genes and see if there are any abnormalities in those chromosomes. And it's kind of cool because in my other job with medical insurance claim adjusting, I see karyotypes from time to time. And sometimes they're normal and sometimes they're abnormal. And it's kind of interesting to see those things in real life examples. Okay, so in this illustration, in this karyotype, can you tell me if it's a male or a female? What do you think? Let's see, does it show it? Guys, there's 2 X's. Is this a male or a female? Someone tell me. We're shy this morning. Okay, it is a female because there's 2 X chromosomes, okay? You definitely need to know 2 X chromosomes is female, X and Y is male. Okay, meiosis is how you produce gametes. Remember, those gametes are egg and sperm. So those sperm and egg, they're formed through meiosis, which is a special type of cell division that helps prevent too many chromosomes from being in a cell. Cells that are produced through meiosis are haploid. This is different than mitosis. The cells that are produced through mitosis are diploid. So meiosis is a special type of cell division that produces haploid cells. This is used in sexual reproduction. So meiosis also occurs in stages, and the stages are named the same way. But there are 2 stages of meiosis. There's meiosis 1 and meiosis 2. Okay, so meiosis starts with the chromosomes duplicating just like it did in mitosis. This happens during interphase. But what makes meiosis different than mitosis is there are 2 rounds of cell division. In mitosis, there's only 1 round of cell division. Okay, big difference, things that you want to keep in mind. So the result of meiosis is 4 haploid offspring cells that came from 1 parent cell. they all would have half the number of chromosomes, so that when they do meet up, an egg and a sperm meet up, they'll form a full pair. So they only have half the number of chromosomes as a result of meiosis. Okay, in interphase, the chromosomes duplicate, just like we talked about before. Chromosomes come in matched or homologous pairs. Remember that prefix homo means self, so these are identical pairs. After interphase, cells go through 2 rounds of cell division, meiosis 1 and meiosis 2. So in meiosis 1, you have those homologous pairs line up. Okay, and just take a minute to look at this illustration, and you can see it on page 90 in your book. Those are, one set is from mom, one set is from dad, okay? So they've duplicated and they line up along the center, and the first round of cell division divides it in half. So you have sister let me see, how do I say this? You have one set of chromosomes from mom in one cell, and one set of chromosomes from dad in another cell. So this automatically does not produce genetically identical offspring, and we'll talk more about that in a little while. Those homologous pairs separate in meiosis 1. You have cytokinesis, and this split into 2. And now you have, this is the end of meiosis 1, those homologous pairs line up and separate. That is what's happening in meiosis 1. In meiosis 2, chromosomes, or those sister chromatins, condense and line up along the center. This is just like metaphase. Yeah, it's called metaphase. It's metaphase 2. You don't need to know the super detailed stages for these. So they condense, they line up. You have 2 cells that are doing this. And you still have your mitotic spindle, pulls them to opposite sides of the cell, and those chromosomes separate. Then you have cytokinesis again. So here you can see those chromosomes being pulled to opposite sides of the cell. This is a stage of anaphase, anaphase 2, I believe. And each of those cells undergoes cytokinesis, and you end up with 4 daughter cells that have half the genetic information that the parent cell had. So you have 4 haploid cells. Okay. There are 3 major ways that you can get variation from sexual reproduction. Because again, the offspring in sexual reproduction are not genetically identical to the parents. They are different. We don't look exactly like our parents. We have features like them, but we are unique. How do we become unique? So first is independent assortment, then random fertilization, and crossing over. These are 3 mechanisms to produce genetic variation. So first, in independent assortment, let's see if I can find independent assortment. This is on page 94 in your book. So in independent assortment, this leads to variation. So because those chromosomes, those have duplicated, this is the very first part of meiosis, they line up, okay, right here. How they're pulled to the opposite sides of the cell, whether it's your mom's chromosomes or your dad's chromosomes, or if it's a mix between mom and dad, is completely random. You just have 1 pair on one side and 1 pair on the other. And it could be a mix of mom and dad's on one side and a mix of mom and dad on the other. So how those maternal and paternal chromosomes line up in meiosis 1 here is random. So if we did some statistics, somebody else did the statistics, I did not. But found that there are over 8 million possible arrangements of chromosomes just through meiosis 1, and that leads to a lot of variation. The second type of variation is random fertilization. And so this is our gametes. Remember, they only have half the genetic information, and which sperm meets with which egg is completely random. And that leads to variation as well, because if we look, this sperm has 8 million possible combinations. The egg has 8 million possible combinations. You combine that, that gives you 64 trillion possible combination in random fertilization. So that leads to a lot of variation. The last type of variation is crossing over. And this is kind of a neat phenomenon that occurs. And you can look at this on page 95. It's a good page for you to look at. It shows crossing over and some illustrations. Chromosomes are able to basically, so you have, there's an illustration that's going to come up. You have sister chromatids that are connected. They can swap genes, and that creates unique new combinations. This swapping of genetic information between chromosomes is called genetic recombination. This happens during meiosis 1. So here, remember in meiosis 1, we have those chromosomes line up along the center, one set from mom, one set from dad. And between these two, you have genes, and they can just swap genetic information so that some of mom's genes is attached to dad's chromosome in multiple places here, okay? They swap genetic information. That swapping leads to even more variation because the chromosome is then not just purely from mom, it has parts from dad, and it leads to a lot of additional variation. This creates what we would call hybrid chromosomes, and the hybrid chromosomes increase variation a lot. So, however, there are, okay, we're going to end in a little bit. Meiosis can have some problems. It does not always go as planned. One of the problems that can occur is non-disjunction, and this is when the chromosomes don't separate properly, where you either have extra chromosomes in a cell or not enough chromosomes in a cell. This can lead to non-viable offspring or developmental abnormality. So normal meiosis versus non-disjunction, and this is on page 96. This is just showing from an ovary and a testis cell the production of gametes. So remember, the gametes are haploid. In fertilization, then you combine genetic information from mom and genetic information from dad, from egg and sperm, to form a diploid zygote. In non-disjunction, something goes wrong. The chromosomes don't separate properly, and either the haploid egg or sperm has additional chromosomes or lack of chromosomes. That can lead to a zygote that has too many chromosomes or not enough chromosomes. So we're going to talk about some examples of non-disjunction. Down syndrome is pretty well known. We also call it trisomy 21, tri for 3. And this is because on chromosome 21, there are 3 copies instead of 2. Okay, and it affects about 1 in 700 children. So there you can see down here, there's the karyotype, and you see there's 3 chromosomes for 21, and there really only should be 2. Okay, some other examples of non-disjunction. Sex chromosome non-disjunction can also occur, and it leads to some different types of disorders. So they have, whether you have too many or not enough, can lead to each own syndrome, separate syndromes. So Turner syndrome is when there is only one X chromosome. So one of the sex cells, either the sperm or the egg, had non-disjunction of the sex chromosomes, and you do not have both pairs. So in Turner syndrome, you can see here, it's lacking the second chromosome in that karyotype. It can lead to webbed neck, like skin that's kind of webby, underdeveloped ovaries. Often the person is sterile, and there can be learning difficulties. I actually had a student last semester who had Turner syndrome. She came up and talked to me about it, and she felt that this was really relevant because we were talking about her genetic disorder. So it happens. And by the way, you can't always tell by looking at someone if they have Turner syndrome. Okay, Klinefelter syndrome. This is where you have 2 X chromosomes and a Y chromosome. So it's almost kind of like a meld between female and male. You have underdeveloped testes, low testosterone levels. Usually they are infertile. They develop breasts. They have female features like more hippie, their hips, long legs, long arms. So they have some female features, but they're technically male. That's Klinefelter syndrome. Jacobs syndrome, or we also would call them super males, have 2 Y chromosomes, and that is when non-disjunction of the sex chromosomes occur in the testes. And these men tend to be taller, they have more acne, the intelligence might be a little stunted, and they tend to be more aggressive, a lot more testosterone. Okay, females or metafemales with 3 X chromosomes, so non-disjunction occurring, whether it's it would be occurring in the ovary. And these metafemales may be sterile. There's no obvious physical abnormalities. You can't tell if someone is a metafemale just by looking at them. They may only find out if they're sterile and they're trying to have offspring, and they get a karyotype done and then find out, oh gosh, I have 3 X chromosomes. That's a problem. Okay, I'm going to stop with this. I want to start on Monday talking about Gregor Mendel. I would go on YouTube and check out the crash course on meiosis. I'm not going to show it today. I think it's what crashed my computer. So check that out. He's a great resource. You will see more from him. I'll have other videos as well. So that is it for today. Remember that all of your labs, all of your Unit 1 homework, which is chapters 1 through 4, all of that is due today, and your exam is due today by midnight. If you are having trouble with your exam, I know it's up, some people have taken it, but if there is some sort of difficulty, text me on my cell phone and I'll get back to you as soon as I possibly can. Remember, my cell phone number is 909-653-5323, and I will be able to respond to you much quicker than my email. I won't be able to check my email until this evening. So if you need to get a hold of me quicker, text me, okay? Any questions about today? Okay. Let's see, did everybody put their chat? Oh, I lost the chat because my computer crashed. I'm so sorry, guys. Let's see. I'll put up an attendance thing today just because I lost the chat. So check for that, and I will make sure you guys get grades for attendance. Thank you so much for coming to class today, and I hope you have a great day. Good luck on your exam, and have a fantastic weekend. I'll talk to you on Monday. Bye.